Hello everyone, I'm Chris Chadwick, your host for another interesting episode of Mac's podcast series, Mac Behind the Trials. Before I introduce my guests, I'd like to remind you that Mac Clinical Research is a global full-service CRO with science at its core. On today's episode, we're joined by Dr. Scott Harrison, Director of Laboratory Services at Mac, to explore the fascinating topic of metabolomics. As part of Mac's comprehensive laboratory services, Metabolomics is an exciting and innovative development in clinical research. So Scott, it would be really useful if you could give us all a brief overview of your role at Mac and the in-house services that you and your team deliver.
Thank you for inviting me, Chris, and it's great to have an opportunity to talk to you today. Within laboratory services, over the last 2 years, we've actually built a state-of-the-art analytical facility in which we can analyze the blood and the IMP or investigational medical products that are part of clinical trials. We've done this as a team effort to the highest possible standards, building quality building it from the ground up with quality at the core. And we've reached out to the regulatory agencies to ensure that we have accreditations that are fit for purpose, to ensure independent quality assessments are made of our work.
Now, that's fantastic. So I have to ask, my first question's got to be in that case, if you could briefly explain what metabolomics is.
So metabolomics is one of the omics sciences. And has its roots somewhere in the 70s. In the 70s, a group of microbiologists coined the phrase metabolome for the small molecule complement of the cells. And then with the other omics sciences, genomics, transcriptomics, and proteomics, it was logical that what we would do is to try to measure all the small molecules. So measure the metabolome. So there's the root of the word, and globally, which is the omic part of the word. So metabolomics is a combination of metabolome and omics. So metabolomics, which then gives us the ability to look at how cells respond at a biochemical level to various challenges, be it temperature, be it if a chemical's added to them, be it how they evolve in their life cycle.
So really microscopic detail then we're talking about.
Yes, it's one of those things. One of the interesting things about metabolomics when you compare it to the other omic sciences is that if we were in this room and suddenly we had the ability to remove all the air and we measured the genome, the transcriptome, the proteome, and the metabolome, the only change that we could actually see would be in the metabolome. So your genome is your potential. Your transcriptome is how the instructions are produced to produce proteins. The proteins are the machines that carry out the metabolism, but it's the metabolism itself that is the true processes of life. The conversion of the fuel, your food, into molecules that the body needs to thrive.
That's perfect. No, thank you very much indeed. Moving on from that, I just wanted to see if you could give us a short overview of its history and ultimately the development of metabolomics.
Yeah, so metabolomics actually really commercially started around the late 90s, early 2000s, where we've had the genomic revolution, we've had transcriptomics and proteomics. And then there was an understanding that we wanted to dig deeper to find out what was really going on at a molecular level in the processes of life. So there were multiple companies that set up around that time frame to look at various things that to prove if if metabolomics could be done, but also what benefits it would give. So, for example, there are metabolomics papers that will allow you to differentiate chemically between a red strawberry and a green strawberry. And I know that sounds superficial because you can just look at them, but it was a proof of concept to be able to show that those differences are echoed at a biochemical level. Similarly with black and brown mice or white and brown mice, or males and females differ chemically. So all these proof of concepts then built up a body of evidence which in those early days of metabolomics, it was actually looked down on by certain parts of the scientific community because we actually were taking a non-conventional approach to science. As science developed, it was somewhat reductionist. What we've trained as scientists to do is break down a problem into smaller and smaller bits and then test those individual hypotheses to see whether or not we could show that they were the evidence supported them or disproved them. Metabolomics took the approach of let's just look at the data that we're going to get and try and see if then we can generate an understanding of how that data gives us information. So it was breaking that normal scientific paradigm of have a theory and test the theory. This was collect data and then create a theory from explain the data.
So a real revolutionary approach then.
Yes, it took many years for it really to establish itself within the scientific realms. And I still remember in those early days facing the challenge of, well, this is just a fishing expedition. And it was quite odd because almost overnight, there was a change in the scientific attitude where metabolomics went from being a fringe science to being an accepted technology. And I don't see how the switch was flicked, to be honest with you. It was quite odd, but it was also quite rewarding for us that have worked in the field for it to gain that acceptance.
Absolutely. So I have to lead on by asking, what brought you into this field then?
So I worked in the scientific industry, in academia before I took a job with a major multinational. And I went to a company in America who was setting up what they called at the time a biochemical processing lab. And that biochemical processing later became metabolomics. But it sparked an interest in me because it was a novel, new approach, a new challenging frontier that we could use a non-standard analytical technologies to push the boundaries of science. And this company, actually, what they were doing was they were knocking out a Arabidopsis genome, one gene at a time. And quite often what they would see is that gene deletion had no visible effect on the phenotype of the plant. The plant grew quite happily, and they couldn't understand why a gene would be there if it was having no effect. And somebody within the company started questioning, so what's happening to the biochemistry? And that's where the metabolomics actually started within that company, because they started monitoring a broad range of chemicals to look at what the effect of those knocked out genes were. And it turned out that there were some genes that had very targeted effects and some genes that had a wild disruption of the biochemistry of the plant, but it was surviving. And then, of course, there were some that killed the plant.
Absolutely.
that's a different story.
So I just wanted to ask if you could tell us the difference between targeted and non-targeted metabolomics.
So this is a debate within the metabolomics community, and it all comes down to the complexity of metabolomics. One of the early resistance to adoption of metabolomics was that metabolomics generates huge quantities of data, and understanding what that data means is actually quite difficult. A programmer friend of mine once said that if you have data plus structure equals information, and then as an add-on to that, information plus structure equals knowledge. So what we did, what the two camps in metabolomics actually argue is that if you go and you measure 10, 15, 100, 200 specific compounds in metabolomics, i.e. you do a targeted method, you can absolutely quantify that number of metabolites from the individual test unknown samples. And that then you can apply that to the metabolic pathways. Whereas what the people who support the non-targeted approach say is, that's all well and good. Yes, you get an understanding of how those specific chemicals are changing within the metabolome, but you're losing an awful lot of information. So the best approach is to look for as much information as you can get, and then dig statistically using bioinformatics to piece together all the significant changes. Because the potential problem with the targeted approach is that you get correlations in changes to a specific condition, be it drought versus well-watered in plants. You can get a correlation which isn't actually directly caused by that. So it can send you down the wrong pathway, whereas the broader non-targeted approach gives you the ability to try and see everything that you can. And then, you didn't mention it, but there's a hybrid of those two approaches, whereas you measure the known compounds that you can measure, but then you also collect all the other data. So you can actually get the best of both worlds, but of course, the overhead, the analytical costs increase because you're doing the data collection effectively for both techniques. So the analytical overhead is larger, and the downstream overheads get larger because it becomes more. The amount of data that you collect can be huge, and that then has knock-on effects with the information that you need to process.
So I've got to ask, in that case, the hybrid version, is it literally called a hybrid version, or does it have a specific title associated with it?
It's one of those that it's the approach that I favor, and I've always called it hybrid, purely and simply because it is the two camps are almost partisan. That you either fall into the supporting targeted metabolomics, or you support non-targeted metabolomics. And my background, I came in through the non-targeted route, but I could see the benefits of knowing what the compounds are. But realistically, does it matter if it depends what you want, what the information you want out of your experiment is.
Yeah, the outcome data is just so important. Yeah,
exactly. So if you want to link back to a specific metabolic pathway, a targeted approach is probably the easiest to understand and the easiest to link back. But if you want to just find a marker for a particular disease or condition, you don't actually need to know, does it make a difference whether it's called Bob or glucose? The answer is not really.
So true. Yeah. Yeah, so true. No, thank you very much indeed for that. So, can I just ask in that case, where does metabolomics fit within the broader landscape of omics fields?
So, as I stated earlier, metabolomics was the latest of the omics sciences. A lot of money was poured into the genome. With the Human Genome Project, we managed to map the entire genome. And then from there, transcriptomics developed with an understanding of which genes were expressed in which area. And those transcripts then are converted into proteins as they're transcribed. Those transcribed proteins, or another way of calling them is enzymes, then metabolize the biochemicals that are coursing through cells and through our bodies. And understanding that at that level is the base level of what the processes of life are. So as a challenge occurs, be it that you give a drug which blocks an enzyme, you can see a shift in the underlying biochemistry where the products that should be produced by that enzyme are no longer produced, or the amount of them are reduced, and therefore it has ripples into the actual underlying biochemistry. And when we look at the root of disease is dis-ease without ease. So what we're moving away from is the normal status of our bodies. The underlying biochemistry changes, which causes problems that the body then needs to react to try and get back to that homeostatic area where all the cells are functioning within normal parameters.
So moving on from that then, can you tell me what are some of the challenges facing this field?
As I mentioned a little bit earlier, metabolomics produces huge data sets. It's based on a statistical approach. So the larger the data sets that you can get, the more information that you can get, that you can tease out of them. Also, the fact that it depends on the strength of the signal. It's relatively easy to differentiate using the sex hormones between a male and a female, for example. But if you're requiring more subtle information, such as what a particular disease is, you need larger data sets, you need more specialized data sets. So you need to understand how to select the correct population to test for a start. Then there's the analytical challenges, the routine chemistry. As I said earlier, our training is reductionist, where we're trained to look at one or two chemicals and give you very precise, very accurate understanding of the levels of those particular compounds. You've got to change your attitude when you're looking at metabolomics, because you're trying to measure everything, and there's a vast array of physiochemical properties that you need to take account of. So one technique might favor more volatile compounds, others might favor more polar or less polar compounds, and we need to be able to fit those all into analytical technologies to get the broadest coverage of that metabolome that we can. Then we move on to, once we've got the data, what do we do with it? There's huge terabytes worth of data that we need to develop methods to process, to check quality, to be able to understand that the differences that we see are not just simply artifactual, but are genuinely caused by the challenge that this living cells that we want to see actually cause. So that requires an understanding in computing and bioinformatics. And then once we processed those huge data sets and we got some information, we need to be able to translate that into what it actually means. So if it's monitoring of a disease, it's is that disease, is that compound actually relevant to the disease, or is it just artifactual through that? If we were looking at depression versus healthy, that depressed people tend to self-medicate, so they have got nicotine in their system, and therefore, if you do not control your system well enough, what you would see is a marker for depression is nicotine. And one example that I can give is on an early trial that I was involved in. Is we were looking at a disease called ALS, or I think in this country it's called motor neuron disease, and we took a population of healthy volunteers, and we took a population of suffering from motor neuron, and we analyzed them, and the data gave us a black and white separation. These people were healthy, these people were suffering from the disease. So we then started looking, and all the markers that we found were simply because those motor neuron people were very ill. We found markers for very ill, which was great, but it didn't give useful information. It was information that you could tell that these people weren't. So
you had to drill down a lot further.
Yeah. So what we ended up doing is looking at other neurodegenerative diseases like MS, like Parkinson's. And what that enabled us to do was get rid of the noise of the general illness to look for specific markers for that particular disease. So this is going right back. You said, what's the challenge? One of the biggest challenges is actually designing the experiment. Even though we're not reductive scientists, we're still scientists. And what we need to do is design experiments that will give the answer to the question. But if we weren't rigorous in our interpretation of those ALS results, it would have been quite easy to publish several papers saying these are markers for ALS, whereas what we had actually produced in the first round were markers for ill.
And that was a really interesting example. I appreciate you sharing that with us. So just moving on from that, I just want to say, how does metabolomics then integrate within clinical research?
So moving on from the example of motor neuron disease, metabolomics can integrate through the drug development process, right from early discovery phase, where you're carrying out your lead optimization. So when you understand that you want to block the activity of this enzyme with your drug. So what you do initially is then add the drug candidate to a culture or an enzyme and show that it's got activity. The next stage of that is to add the potential drug molecules to cell cultures and see what other effects it has. At that point, you can use metabolomics to show that the drug or the molecules that you're selecting, which one of those is actually having the best effect. And the best effect traditionally was that it would bind to the enzyme with the highest affinity. The trouble with that approach is that it doesn't take into account what we call off-target effects, where it also binds to other enzymes and causes blockages of other pathways that you're not trying to target. That is what is one of the causes of what we know as side effects. So we had a experiment that we were carrying out, which was for a major pharma company, and they had, I think it was about 10 drug candidates, which they wanted to test in a cell system where we could look at the metabolome of those cells and how their drug candidates actually worked. And the usual doctrine is that you give the most active compound, that is your best drug candidate. What the metabolomics experiment showed in this case was actually the least active drug candidate was the best drug. And the reason why I say it was the best drug is it had the least off-target effects. So it disturbed the metabolism the least. So basically what was happening is, yes, you would have to give more of it, but there is less likelihoods of side effects, and therefore the toxic effects, which we all know is one of the major causes of drug failure through the pipeline. And then, of course, what we can do is moving further down the pipeline. We also know that individuals react differently to drugs. And this is one of the big challenges in clinical trials, is that an individual, I might metabolize a drug very quickly, you might metabolize it quite slowly. But how do we know that other than if we've given you the drug? What we can do is use metabolomics almost as a screen, so we can work out what group you fall into with prior knowledge, so that a drug company can select that I don't go on a trial because my body gets rid of the drug too quickly, so it can't possibly.
So it's really moving towards personalised medicine there,
Exactly,
isn't it?
yeah. This personalised medicine has been one of the goals of the medical industry for many, many years. And I was thinking about this on my drive to this meeting, and I was thinking, has personalised medicine ever come about? And the answer is, yes, it has. Both you and myself wear glasses. We have personalised medicine. I can tell you if I gave me your glasses and vice versa, we probably wouldn't be able to see. But we've got our own prescription. And what metabolomics can do is empower that personalised medicine to make sure that we get the right drug to the right person. And we talk about diseases. Classically, what's happened with disease is that they've been classified under symptomology. So we take a common disease like asthma. The general public will assume that asthma is asthma is asthma. If you start digging into the literature, there is no commonly accepted figure, but somewhere between 7 and 20 different forms of asthma. Each of those different forms of asthma requires a different treatment. But what happens when you go to your GP, you're given an inhaler.
There's your prescription, and that works for everybody.
Yeah. Wouldn't it be great if you could go to the doctor takes a blood test and goes, you've got this form of asthma, this is the pill that you need to take, and it's more efficient, it saves money, and it leads to better outcomes with patients ending up with the outcome that they need sooner.
Absolutely. And what great patient satisfaction you get on the back of that as well. So I want to just talk to you about the concept of patient stratification, if we can.
Yes, of course. So as I was mentioning, every individual is an individual, but we fall into groups. These groups have different metabolic profiles, which we call biochemical phenotypes. Those biochemical phenotypes are either suitable for a particular drug treatment or not suitable for a particular drug treatment. And by understanding that early on, we can actually end up with a targeted clinical trial which is less likely to fail, has better outcomes, is more efficient. And by choosing those correct groups for the trial, it enables us to stratify, as we say, the patient population and give the drug company or the sponsor the information that they need ongoing to enable this future personalized medicine as well, where again the drug is not just being prescribed. And a figure that I've heard bandied about but don't have the evidence for is that 80% of drugs that are prescribed are not actually useful. What we would like to do is get it to 80% of the drugs that are prescribed actually work.
So, can I ask now then, does it mean that we can actually accelerate participant recruitment in clinical studies?
Potentially, the answer to that is yes. In the fact that we can understand the biochemical phenotype, it means that it enables us to collect the data and select the correct individuals for the trial, so that we're not then in the number of people that we need on the trials potentially can be reduced, because we know that we're reducing the noise that's in the data, but we're also making it more targeted, more suitable for the actual requirements of the sponsor. So it's better for the participants because they're not taking drugs that that aren't good for them. It's better for the sponsor in that they're getting less noisy data, and it's therefore improving the speed and efficiency of those clinical trials.
No, that's perfect, and that's exactly what I want. So I'd be grateful for your vision in that respect for the future of metabolomics, and how does it align with the goals of personalised medicine, which we touched upon a little earlier?
Yeah, so I truly believe that the way forward, we're constantly hearing in the news in many countries across the world the strains that health systems are under, the expectation of people that they will be treated for their disease quickly, efficiently. And I feel that metabolomics is on the cusp of being adopted as a common tool within clinical practice, in that by understanding the metabolic phenotype of an individual, you can then help guide the treatment that they get, so that they get the right treatment, the right time, increasing the efficiency, reducing the burden on the health systems, but also potentially reducing polypharmacy problems where people are taking multiple drugs and those drugs start interacting. So by having an understanding of how that individual's body actually works, it potentially enables the right drug to be prescribed, right drug first time, every time.
No, fantastic. Now, you touched upon a little earlier about phenotype testing, and I just wanted to see if you could, your view on the fact that how far away it is from general practice.
That's a very interesting question, because as I mentioned previously, all health systems are strained. So although this technology, I truly believe, is applicable to that type of environment, I think there'll be a resistance, and the resistance is from the other competing technologies, the scanners, and also the conservative nature of the medical industry itself. Things have to be proven. I think that the weight of data is coming to support the adoption of a biochemical phenotyping within clinical practice, as I mentioned. But what that will probably occur through the final adoption will be as a result of companies like ourselves using those phenotypes in clinical trials, where we can show that a specific phenotype benefits from a new drug. Then that acts as almost like a companion diagnostic, which enables the drug company to say, if this person has this profile, they need our drug, and therefore they got a method of actually supporting both the beneficial effects and helping support the efficacy of the drug as well.
No, that's fantastic. So you'll be pleased to know my penultimate question for you. So I just want to know how metabolomics is transitioning from research-focused applications to commercial uses.
Yeah, and that is a very interesting point. As I mentioned, I think right up front, it's been an interesting journey in metabolomics. Because metabolomics was made popular or raised in the scientific community by both commercial entities, but also by academics. And there was a lot of money pumped into academia to support metabolomics as a research tool. And in the UK, we had the National Phenome Centre down in London. We had the Birmingham Phenome Centre, which I helped set up in Birmingham. And all of those have been useful in an academic sense, and have led to this almost acceptance of metabolomics as a tool. And the next step is for companies like ourselves to actually take it from the scientific interest to use it in a real world, a real study environment where we can show the benefits of the metabolomics, not as an academic exercise, more as a benefit to give information that couldn't be achieved in any other way. And that's one thing I've been very happy with the vision of the exec within MAC, is that they can see that metabolomics has potential to do exactly what our declared aim is, which is improve the scientific rigor, the efficiency of clinical trials, and shorten the time it actually takes to get that supporting data that we need to get to prove a drug is safe.
Absolutely. Now, you touched upon there about executive insight within MAC. So it leads on nicely to my final question. I just want to know, is MAC currently the only CRO, in your view, that's incorporating metabolomics into clinical trials?
To the best of my knowledge, the applied metabolomics that MAC is offering its sponsors and in discussion with is the only use of metabolomics in real-world clinical trials. Of course, there's been trials in the area of discovery area, which I've talked about 2 or 3 earlier in this conversation. But in a real-world clinical trials, MAC is unique, and again, it's because of the desire to ground all our trials on hard scientific rigor, so that we can make sure that our sponsors, the trials that they conduct, give the information that they need to prove both the efficacy and safety of their molecules, of course, which they've invested a lot of time, effort, and money in. And this potentially can save them money by making the trial more efficient.
No, that's fantastic. Well, I just want to conclude in that case by saying thank you, Scott, for your time and sharing your knowledge and expertise in us. And more importantly, giving us a comprehensive outline of what metabolomics is, and also the potential, as far as its role is concerned, in personalised medicine, which was particularly interesting from my perspective. So, as well as Mac being amongst the first companies to implement it in clinical research, which is fascinating. So, just in concluding and thanking Scott once again for his time, I just want to make sure that you subscribe to all of our channels to stay updated on all the upcoming episodes within our podcast series. And also, don't forget to follow us on LinkedIn for even more insights moving forward. Thank you.
Thank you.